Okay, thank you very much, Misha. And I know we are on TV, so this is our way to improve our connection with the scientific community. What I will do today is try with my talk to really introduce the symposium. I will try to show you a little bit of what we have been doing on early experiences, and I will touch upon the importance of early environments and how these can interact with genetic susceptibility to possibly lead to increased risk for psychopathology. We have been working in this field for many years now. We know from a number of epidemiological studies in humans that trauma, neglect, a poor socioeconomic environment can have a number of disruptive effects on the development of the nervous system and lead to an increased risk for developing psychiatric disorders at adulthood. We also know that a lot of this is mediated through the mother-infant interaction. Now, this doesn't mean that fathers don't also have a very important effect, but the kind of mutual regulation that we see very well, for example, in the rodent, and that occurs between the physiological systems of the mother and the physiological systems of the infant is it tells us something about what the mechanisms, what the underlying mechanisms of these long-term effects might be. So we believe that there is actually a regulation that goes on between the mother and the infant in terms of arousal and in terms of those pathways, those neurochemical systems that regulate arousal, response to stress, neuroendocrine responses to stress at adulthood. And so this modulation ultimately results in a good setting of the system. If something goes wrong, the system is set either too high or too low, and this might result in an increased risk. In particular, we have to think that what might occur is that exposure to stress early on might modify these systems in a way that when stress comes at adulthood, and all of us are exposed to stress sooner or later, then that might precipitate pathology. We know, in fact, that depression, major depression, and many other psychiatric disorders, they arise at times when there is a lot of stress. Think about adolescence. It's a time when young people are experiencing a lot of stressful situations, maybe because of they are not accepted by the community, or they just undergo a lot of changes in terms of their hormonal systems. All of this becomes a second hit that can precipitate psychopathology. And of course, this is more likely in individuals which have a genetic susceptibility. So sad. We really know very little of which are the biological mechanisms that underlie these long-term effects. So what is really happening in the brain? We have to hypothesize that those circuits which are sensitive to stress, and you have seen from Bruce McEwen's talk, and many others, that one of the important brain areas which are very sensitive to stress is the hippocampus. And in particular, we have seen how the hippocampus, the dendrites, can shrink or return to their normal size depending upon exposure to stress and glucocorticoids. There are a number of regulators of this system, and we have always been interested in neurotrophins, and among these NGF and BDNF, because they can regulate these circuits, and they are also involved in activity-dependent activity of the CNS. We know that these neurotrophins, and here I'm showing you some papers that relate to NGF levels in humans, in the peripheral circulation, And what you can see is that there are a number of stressful situations where these neurotrophins are increased in humans. For example, when we decide to quit smoking, or if we are caring for a relative which is ill. I'm thinking of someone who's caring for an Alzheimer patient. That's a very stressful situation, and it has been shown to increase NGF levels in the circulation. For BDNF, you probably are more aware of the research that has shown how it is implicated in depression. There is a reduced, consistently found reduced serum BDNF level in depressed patients, and antidepressants can restore levels to those of normal controls. Not only that, but we know that, for example, in bipolar patients, if we subdivide the population according to whether they have had traumatic life events, we see that those who have experienced trauma early on are more likely to show reduced levels of BDNF. So there is a very important interaction between early experiences and psychopathology, and BDNF is involved in this mediation. So this summarizes what I've told you so far. So that really in addition to other systems, neurotrophic factors should be seen as potential mediators of the effects of early experiences on later psychopathology. And of course, the changes that we see might be regulated epigenetically. We have shown many years ago, for example, using an animal model of trauma, maternal separation in this case, maternal separation stress performed for 2 weeks, 3 hours every day from postnatal day 2 to postnatal day 14, results in very different effects early on and later during life. This is telling us two important things. The first is that when we test an animal at the time of trauma, we see some neurobiological effects which are very different from what we might see later on at adulthood. And so we really have to follow the whole developmental curve to have a very clear idea of what's going on. In particular, what we see here is that right after these multiple separations, BDNF levels are increased in the hippocampus and in the prefrontal cortex. But if we look at an adult animal that has had the same maternal separation, actually what we see is a profound reduction in the expression. And of course, this reduction is very consistent with what we know about depression in humans, where BDNF levels are found to be reduced. Now, we were not really satisfied in working just with rodents, because, of course, rodents are a very good model. They are very well characterized. We know very well maternal behavior. We know a lot of literature about long-term effects of early experiences. But as I was telling you at the beginning, we are really looking into complex interactions going on during early postnatal life. The complex interactions that we see in humans, for example, the type of exchange, of social exchange between a mother and an infant are clearly not the same that we can see that we see in a rodent. So we had the privilege of being able to collaborate with Steve Sumi at NIMH in Poolesville, and he has this very nice model. I thought we really have to try and collaborate with Steve, because you must understand how this is a really unique model. There are other non-human primate models of maternal separation like the one he has. And so also from an ethical point of view, we really have to sort of gather all the data that we can from this model. And now what he does is he has a very nice colony of rhesus macaques. Some are mother-reared, some are reared with same-age peers. What happens is that in the absence of the mother, the same-age peer, of course, compensates for a lot of social interactions, but it's not like a mother. So all the attachment that goes on between the mother and the normal mother and an infant is really becoming here an anxious attachment with the peers. So we have a situation that at adulthood would lead to animals which are more likely to become subordinates and that have a lot of problems in their social behavior. Now we looked in this model, of course, in the peripheral circulation, whether levels of neurotrophins like BDNF might be modified as a result of this experience. So the experience of being reared with a peer, with the same age peer. What you can see here is that basically we find, first of all, a very nice sex difference. Females have higher levels of BDNF than males, and this is something that recapitulates also the human situation. In humans, females have higher levels of BDNF in the blood than males. But also what we see is that at 2 months of age, which is a good time when they, a long enough time when they've experienced this peer rearing, the females are responding to this situation with increased levels of BDNF in their circulation. Now we knew that from the human literature that there are genetic variants in the BDNF gene, and we were wondering whether we could find a gene bioenvironment interaction in this effect. So in other words, first of all, whether we could find a polymorphism in the macaques. We didn't know whether there was, and whether it was similar to the human polymorphism, and whether this might actually interact with the condition. We actually were able, thanks to the collaboration with the laboratory of Peter Lesch, we found a very, very similar SNP to the one that is found in humans, which is a substitution of a valine with a methionine. And what we found is that now if we look at our data, dividing the animals depending on their genotype, these are the animals that have the val-val, and these are the animals that have the met-val or the met-met. You see that here again we find this increase in BDNF levels in the circulation as a response to peer rearing. But now the animals that have the SNP, both the heterozygous and the homozygous, which are actually very, very little, you don't see anymore this response to separation. Now the way we interpret this is that the increase in BDNF, very much like what we saw in the rodent, is probably a response to the stress of the situation in which these animals are posed, and probably in the subject that have the SNP, this response is prevented, and this is of course a maladaptive thing because whatever BDNF is doing is trying to compensate for the lack of maternal experience. Now, of course, we know very well that when we find differences in gene expression, there might be an epigenetic change going on. And in fact, we hypothesize that many of the effects, of the long-term effects of early experiences that have been documented in the literature since 40 years now, are probably due to changes in the way the genes are transcribed due to changes in their methylation or their acetylation. So one possibility is that, in fact, what we see is are long-term effects of this, of changes in epigenetic changes in the gene. We have now started through a consortium with other groups, other European groups, and together with a Canadian group, the Moshe Zif Lab, a very large study to actually look at epigenetic changes as a result of early experiences. This project is called Poseidon, and it really wants, in a very translational perspective, wants to put together rodent studies, non-human primates, and human offspring, and trying to find an epigenetic signature that can tell us really what these experiences with these traumatic experiences are doing to the expression of genes. And in particular, in addition to BDNF, of course, the other candidate gene system is the glucocorticoid receptor, because we know from a great amount of literature that the effects of the glucocorticoid system is very much affected by stress.